Assalamualaikum and hi. We are from Group 6. We'll present to you about enzyme pectinases. Group 6 consists of Nur Izani, Nur Najiha, Nur Shafiqah, Nurul Farisha, and Nurul Nuha.
Hi, my name is Nur Nabati Mumayyamin. I am going to present the definition and structure part. Pectinase is the enzyme that can break down the pectin, which is polysaccharide substrate that can be found in the cell wall of plant. Pectinase also can be referred as pectin enzyme, which including pectolase, pectozyme, and polygalacturonase. Next, pectinase has a structure of high molecular weight heteropolysaccharide that made up of alpha-1,4-linked galacturonic acid residues. The side chain of pectin molecule consists of aramnose, arabinose, galactose, and xylose. Pectinase can be classified into pectinasterase, protopectinase, and depolymerizing enzyme, which are hydrolyzing glycosidic linkage includes polymethyl galacturonase and polygalacturonase, while cleaving includes polymethyl galacturonate and polygalacturonate lyase. The classification of pectinase is according to pectin, pectic acid or oligodegalacturonate, and pectinase at by-trans elimination or hydrolysis, where the cleavage is random or endwise.
My name is Nur Izani binti Mazlan and I'm going to further present about the history of pectinases. Once upon a time, there was a scientist named Henry Brackenwood. He was the first man in the land to discover a structure called pectin, and in 1825 he isolated and described pectinase. Thanks to his discovery, many developments in the industry have been made. The first commercial use of pectinase was in 1930 to extract juices, and then in 1960s it started to be used in the wine production as well. Pectinases are now an integral part of fruit juice and textile industries, as well as having various biotechnological applications. A few of the major applications include extract juices, like I mentioned, wine production, fatty wastewater treatment, paper making industry, oil extraction, and coffee and tea fermentation. The enzyme can be found in plants, bacteria, yeast, and fungi, although fungi is the most preferable as the source of pectinase. The most popular fungus used to obtain pectinase is Aspergillus niger. It's also naturally produced by organisms like insects, nematodes, and protozoans. Aspergillus species, Erwinia species, Bacillus, and Penicillium species have been extensively used in commercial production of pectinase. A little insight on the recent prospect of enzyme production in the industry is that enzymes comprise a well-established global market projected to reach 6.3 billion USD in 2021. Studies have also reported that pectinase of microbial origin accounts for 25% of global food and industrial enzyme sales, and their market is increasing continuously. That's all from me.
Hi, my name is Shafiqah and I'll present to you the processing method in the enzyme production. Enzyme splits polygalacturonic acid into monogalacturonic acid by opening the glycosidic linkage, soften the cell wall, and increase the yield of juice extract from the fruits. Two major sources of the enzyme pectinase are plant and microorganisms. Solid-state fermentation will mainly used in the enzyme production because the potential advantages such as simplicity, high productivity, and concentrated product over submerged fermentation. This is the example of the common process that used in the industry. The pectinase enzyme will be added after the heat exchanger and undergo the process until centrifugation. Then the pectinase enzyme will be added again along with the amylase enzyme and undergo the process until the ultrafiltration. There are three steps in pectinase enzyme production. First, culture condition for pectinase production, where we decide the substrate necessary inoculant temperature, fermentation, time, pH, and aeration. Moisture content was maintained up to 65% in solid state for fungus growth for production of enzymes. The second step is extraction of culture filtrate, where at final stage of this step, the extracted solution will plugged with cotton and preserved at 40 degrees Celsius with 1 drop of toluene. And the last step is protein estimation and pectinase assay, where we measure the reducing sugar of extracellular enzyme. This is the important parameter in pectinase production. The temperature is important to determine the success of solid-state fermentation. Carbon source will improve the growth of the fungus and enzyme production. It needed to maintain the moisture content up to 65%. Lastly, effect of pH. Low or high pH value will inactivate the enzyme and may affect production.
I am Farisha and I will proceed with the mechanisms involved. As we know, the pectinolytic enzymes are protopectinase, polygalacturonases, lyases, and pectinasterase. The pectinases degrade pectin by reactions of depolymerization and deesterification. The figure shown in the slide is the mechanism of action of pectinases. As we told before, there are two types of mechanism involved, which is hydrolyzing and cleaving, which for the hydrolyzing glycosidic linkages, there are two types of enzyme involved, which are polymethyl-galacturonases, which catalyze the hydrolytic cleavage of alpha-1,4 glycosidic bonds. They may be endo causes random cleavage of alpha-1,4 glycosidic linkages of pectin, preferentially highly esterified pectin, while for the azo causes sequential cleavage of alpha-1,4 glycosidic linkage of pectin from the non-reducing end of the pectin chain. Second one is polygalacturonases, which catalyze hydrolysis of alpha-1,4 glycosidic linkages in pectic acid, polygalacturonic acid. They also have two types, which for the endo one, also known as poly-1,4-alpha D-galacturonide glycanohydrolase, catalyze random hydrolysis of alpha-1,4 glycosidic linkages in pectic acid. And for the azo one, also known as poly-1,4-alpha D-galacturonide galacturonohydrolase, catalyzes hydrolysis in a sequential fashion of alpha-1,4 glycosidic linkages on pectic acid. The figure shown is polygalacturonases hydrolyze the O-glycosyl bond in pectin's polygalacturonal network, which resulting in alpha-1,4 polygalacturonic residues. For cleaving enzymes, cleaving alpha-1,4 glycosidic linkages by trans-elimination. There are two types of enzyme involved, which is one, polymethyl-galacturonate liases, which catalyze breakdown of pectin by trans-elimination cleavage. They have two types, which is endo and azo. For the endo one, also known as polymethoxygalacturonide liase, catalyzes random cleavage of alpha-1,4 glycosidic linkages in pectin. For the azo one, catalyzes stepwise breakdown of pectin by trans-eliminative cleavage. For the second one is polygalacturonate liases, which catalyze cleavage of alpha-1,4 glycosidic linkage in pectic acid by trans-elimination. They also have two types, which is endo, also known as poly-1,4-D-galacturonide liase, catalyzes random cleavage of alpha-1,4 glycosidic linkages in pectic acid. For the azo one, also known as poly-1,4-alpha-D-galacturonide exoliase, catalyzes sequential cleavage of alpha-1,4 glycosidic linkages in pectic acid.
Hi, my name is Nur Najiha binti Zainon. I'm going to present advantages and disadvantages part. First, I will highlight the health benefits and uses of pectinase. Pectinase is a valuable digestive aid since pectin is a major component of human diet. It helps to improve the nutritional value of the foods. The capacity of pectinase is to break down pectin and give it several unique benefits. These benefits mainly affect the digestive system, including support for intestinal bacteria, seasonal support, and colon function. Pectinase can help to reduce the intestinal absorption of certain food materials that may be due to seasonal conditions. Furthermore, pectinase can help to promote a healthy level of bacteria in the intestines by maintaining the optimal pH range for their growth. These bacteria are important to effective food digestion. Pectinase can help with digestive support by increasing the bioavailability of sugar in foods. Lastly, pectinase can improve colon health by providing the energy that is required for the proper operations of the colon. The pectin breakdown will produce fatty acids such as butyrate. It is required to maintain a healthy degree of intestinal motility. I'm going to further present about the solid-state fermentation. Solid-state fermentation we use for pectinase production. It is characterized as a fermentation process that is carried out in the absence or near absence of free water. The advantages of solid-state fermentation. Firstly, solid-state fermentation is a low-cost media. Recent advances in industrial biotechnology offer a range of opportunities for the use of low-cost agro-industrial waste in solid-state fermentation for the production of pectin enzymes using fungi. The use of solid-state fermentation benefits in the simulation of their natural environment, which helps in improved adaptation to medium and higher enzyme production. The reduction of microbial contamination due to lack of free water and the possibility of obtaining concentrated enzymes, as enzymes can be extracted with a limited amount of water. Lastly, solid-state fermentation has a less chance of contamination due to lack of free water. Next, the significant drawbacks in the development of these enzymes through solid-state fermentation involve the agitation frequency. Agitation is required in solid-state fermentation to remove the heat produced during the fermentation process and to mix the oxygen transferred to the solid medium. The pace of agitation in the solid-state fermentation is very sluggish. It is recorded from 1 to 3 times a day. The agitation of the medium may change the nature of the particle to fungal mycelium. So, agitation is a significant obstacle to the successful application of the solid-state fermentation. Next, difficulties on scale-up. Scaling up the solid-state fermentation method and estimating biomass has been a major challenge. A variety of bioreactors have been developed to solve the problems of scale-up and to a large degree online monitoring of many parameters. These bioreactors also monitor heat and mass transfer, which are difficult to handle in the simple fermentation system. Lastly, the advantages of solid-state fermentation is mixing nutrients not uniform. The mixture of nutrients using this method is not uniform, thus it will produce higher impurity of the product.